Good afternoon, everybody. Thank you for having me here. Today, I'm going to talk about the taxonomic classification of spiders based on image texture analysis using multifiltering. So the biodiversity classification remains a challenge due to the immense volume of undescribed species and limited resources to do so. So for example, if we think about arthropods and invertebrates, we actually know 1/5 of the estimated biodiversity that is out there. This is our current limitation on this problem is time, because we use tools as morphology or DNA that help us a lot to classify biodiversity, but that they are slow tools. So the general objective of this work is to investigate and validate machine learning methods using image texture analysis and multifiltering for the taxonomic classification, in this case, of spider families. And what we want to achieve here is to at least accelerate species identification, or maybe give another tool to do so. And we hope that many natural history museums use it as other type of biologists. So which was our methodological framework for this work? We based it on a work of Contreras on a database of fingerprints. And they did this following arrangement. They did data argumentation, image processing, image representation, and finally classification model definition. So we did something similar in this work. So to start, we processed like 25,000 images. And we did argumentation of them on with adding 6 different images per image, 6 different treatments. And this data argumentation, as you may see here, we have the original image. Then we flip this image in all the possible ways. And then we resize the image. And finally, we did a noise addition to the image. And all this data augmentation was with the purpose of reducing the overfitting of the model, and of course, to increase the generalization of it. Continuing, we did the image processing. And in this set, we did 4 filtered images, and then we equalized them at the same time, obtaining more or less 100,000 images on our dataset. So what we did first in the image processing, we did a region of interest, or what we call ROI2. So we have our original image. Then we did binary masks, Fisher mask. Then we apply contours to delimit the features that we were looking for. Then we apply a final mask to obtain finally the ROI image. And as you may see, this processing was to reduce the background noise. So in the spider images, you may see that now we have the spider in the image of ROI. That is the thing that we want to compare, and not the background that is full of noise. Okay. So in our work, different to Contreras, we apply contours and final mask as a new contribution. So we apply new tools to increase or improve our image processing. Continue with the image processing, we also, as I said, we did filtering and equalization by applying multifiltering. So the filters images you have in the first row, and in the second row, you have the equalized images of those filters images. And you can see that we have the original images, then apply Gaussian, guided, and Laplacian filtering, and then to all of them equalize it. And what we want to achieve here was to highlight the textures that were important from the images for the model, so it could classify them in the ways that we want. And in this case, we also have a new contribution with respect to Contreras, because we use color images, and so we equalize them using the lab space coloration that improve the images using natural colors, or improving the natural colors of the images. Continuing with the methodological framework, we did image representation, and in this part of the system, we actually transform the pixels into vectors. And I'm going to tell you more specifically what we do. So in one hand, we have the original images. To these original images, we apply different type of descriptors that transform the pixels into vectors, vector information, and we apply different descriptors like SIF, then SIF, and bricks. But we did the same to the filter images and also to the equalized images. So then that's why we have 3 times the number of vectors from each image, totalizing 3,000 more or less vectors. And then all these vectors, we concatenated into a final vector concatenation to have this big vector per image that is all the information from that images. So in the image representation, we need to map this information in a simpler way, because this final vector has a lot of information. So what we did, if you're thinking about a spider image, after applying a descriptor like shift, we are going to have a description matrix that is the one that I'm showing here. And remember that we apply different types of descriptors to each image. And this description matrix can traduce itself in a final vector. But this is really huge amount of information. So what we did here, applying Contreras' framework, was to map the measurements statistics. So we did the mean, the standard deviation, the entropy, the squeeziness, and the kurtosis of the final vectors, and obtain all these statistical values. So that's why we, from now to the end of the talk, we are going to talk about the descriptors as a statistical descriptor. That is the S-SIF, S-densif, and S-bricks. And as we did before, we for the final vector that we analyzed was the statistical vector concatenation. So all these mapping measurements from the description matrix, we transform it in a statistical vector concatenation that we study at the end. But we did not only this. We also test 7 different versions of combinations of descriptors by themselves and also in the total combination of all of them. Okay. Okay, finalizing the methodological framework, we did a classification model definition. And for that, we apply a dimensionality reduction using singular value decomposition that we apply from a matrix of 10% to a matrix of 80% of the entire datasets. Also, we apply data normalization, vector normalization using min, max, score, and robust normalization. And finally, we trained our classifier using support vector machine, using different type of kernels like linear, RBF, or polynomial. As we did in other cases, we also apply new contributions in this part of the framework, utilizing several kinds of kernels that, in contrast, they only use a linear kernel for the analysis. So our results, the classifier configuration. So we studied 3 types of targets. One that is the family that has 34 categories, and structures of the spider, like genitalia, for example, that there were 8 categories. And finally, we have a binary category that was if the specimens were female or male. Our data set was not balanced whatsoever. It has a lot of missing data. So we, after the first the preliminary analysis, we need to do class exclusions. So, for example, for those categories in the family target that has less than 60 samples, we had to remove it from the analysis because they were not doing okay. So we ended up with 10 families as categories. And for the structures category, for the structure targets, we ended up with only 3 categories. So to also study or to balance the data sets, we did resampling using SMOTE, that is synthetic minority oversampling technique, that improved the minority class coverage. And also we apply weightings on the matrix using balance, and we test both of them independently to see which one of those work better in our dataset. And well, this was new also on a new contribution, comparing with Contreras, because the data matrix that they used, the data set was totally complete and clean, and it was not the case in our data set of spider images. And well, we apply the classic metrics for this type of approaches, like accuracy, precision, recall, and F1 score. And the last one was the one that we use as was also used in similar works as ours. So our results in the target variables, the family that is the higher granularity target, we have to use, the best scenario was when we used 2 different descriptors. The dimensional reduction was until 80%, so 80% of the entire data matrix can explain the data. The normalization was its score and the kernel RDBF, but we have a F1 of 0.39. That is really low, but it's expected for a high granularity variable. Target. The structure that was our intermediate granularity target, the best scenario was when we use all the 3 descriptors in combination. Also, something curious that the dimensional reduction, the 20% of the data matrix could explain these results. Normalization and kernel was the same as the before, and F1 was 0.76. That is a really interesting value. We were really happy about this. And our low granularity target, that was a binary one. We also, the best scenario were when we apply at least 2 different kind of descriptors. The dimensional reduction was 80%. The normalization and kernel was the same as the other 2. And the F1 was also high for our expectations, 0.7. So making a summary about this, the best and worst cases scenarios in all our analysis and comparison, the best scenarios were those when we apply a score normalization and kernel RDF. And also when we apply a mode, much better than when we apply a balanced matrix. And the worst case scenario was all the time when we use only one descriptor and any kind of other type of normalization or kernels. So concluding my talk, our model was we built a robust pipeline for spider classification. We obtained high performance for low and medium granularity targets. We introduced a lot of new contributions comparing with our previous work. Well, of course, we have high granularities. The high granularity targets, they remain challenging as expected. When we compare our work with previous works, we found that all the previous works, they focus on low or medium dataset granularities when they study insect images or other spider image database. And they work with low or medium data granularities. They never did a high granularity test. Also, in all these cases, they use single descriptions only when we explored a lot of combination. And we also found that if we apply more combination of descriptor, we have better results, okay? Also, in the past, they never use C-score normalization or even SMOT. We compared both, and we found that it's really important to use both of them for at least our data matrix. And we expect that this could be true for other data matrices used in the past. And as I told before, also in the past, they only operate in lower diversity datasets. That in our case, it was really high, the diversity of different tags that we were analyzing. So finalizing my talk, in our perspective, we would like to expand our datasets. We have other colleagues that could share the spider data images, and we could do a new analysis. And of course, to explore all our data compared with neural networks. So without further ado, thank you very much, and see you in the next time.